Hey guys, welcome back to another episode of my channel. We'll be going over concept 6.3. The eukaryotic cell's genetic instructions are housed in the nucleus and carried out by ribosomes. So, on the first stop of our detailed tour of the eukaryotic cell, let's look at the two cellular components involved in the genetic control of the cell: the nucleus, which houses most of the cell's DNA, and ribosomes, which use the information from DNA to make proteins. So, for my notes, nucleus contains most of the cell's DNA. Excuse me. Excuse me. Okay. Ribosomes use instructions from DNA to make proteins. The nucleus. Information central. The nucleus contains most of the genes in the eukaryotic cell. Some genes are located in the mitochondria and chloroplast. It is usually the most conspicuous organelle. See the purple structure in the fluorescent micrograph. So, this here. Averaging about 5 micrometers in diameter. The nuclear envelope encloses the nucleus, separating its contents from the cytoplasm. The nuclear envelope is a double membrane, so two membranes. The two membranes, each a lipid bilayer with associated proteins, are separated by a space of about 20 to 40 nanometers. The envelope is perforated by pore structures that are about 100 nanometers in diameter. At the lip of each pore, the inner and outer membranes of the nuclear envelope are continuous. An intricate protein structure called a pore complex lines each pore and plays an important role in the cell by regulating the entry and exit of proteins and RNAs, as well as large complexes of macromolecules. Except, at the pores, the nuclear side of the envelope is lined by a nuclear lamina, a net-like array of protein filaments. In animal cells, these are called intermediate filaments. Remember in the last video, plant cells don't have intermediate filaments. And these array of protein filaments that maintain the shape of the nucleus by mechanically supporting the nuclear envelope. There is much evidence for a nuclear matrix, a framework of protein fibers extending throughout the nuclear interior. The nuclear lamina and matrix may help organize the genetic material so it functions efficiently. Within the nucleus, the DNA is organized into discrete units called chromosomes, structures that carry the genetic information. Each chromosome contains one long DNA molecule associated with many proteins, including small basic proteins called histones. Some of the proteins help coil the DNA molecule of each chromosome, reducing its length and allowing it to fit inside the nucleus. The complex of DNA and proteins making up chromosomes is called chromatin. When a cell is not dividing, stained chromatin appears as a diffuse mass of in micrographs, and the chromosomes cannot be distinguished from one another, even though discrete chromosomes are present. As a cell prepares to divide, however, the chromosomes form loops and coil, condensing and becoming thick enough to be distinguished under a microscope as separate structures. Each eukaryotic species has a characteristic number of chromosomes. For example, a typical human cell has 46 chromosomes in its nucleus. The exception are human sex cells, eggs and sperm, which only have 23 chromosomes. A fruit fly cell has 8 chromosomes in most cells and 4 in the sex cells. A prominent structure within the non-dividing nucleus is the nucleolus, plural nucleoli, which appears through the electron microscope as a mass of densely stained granules and fibers adjoining part of the chromatin. Here a type of RNA called ribosomal RNA, rRNA, is synthesized from genes in the DNA. Also in the nucleolus, proteins imported from the cytoplasm are assembled with rRNA into large and small subunits of ribosomes. These subunits then exit the nucleus through the nuclear pores to the cytoplasm, where a large and small subunit can assemble into ribosome. Sometimes there are 2 or more nucleoli. The number depends on the species and the stage in the cell's reproductive cycle. The nucleoli may also play a role in controlling cell division and the lifespan of the cell. As we saw in figure 5.22, the nucleus directs protein synthesis by synthesizing messenger RNA, mRNA, that carries information from the DNA. The mRNA is then transported to the cytoplasm via nuclear pores. Once an mRNA molecule reaches the cytoplasm, ribosomes translate the mRNA's genetic message into a primary structure of a specific polypeptide. Remember, the primary structure of a polypeptide is the string of amino acids. The string of amino acids that are the primary structure of creating a protein. Okay? So, for my notes here, I wrote, nucleus contains most of the genes. Genes are also found in mitochondria and chloroplasts. Nucleus is enclosed by a nuclear envelope, a double membrane that separates the nucleus from cytoplasm. 2 lipid bilayer membranes separated by a space of 20 to 40 nanometers. Pores or holes, with holes like within the structure, that are approximately 100 nanometers in diameter. Pore complex is a protein structure that line each pore and regulates entry and exit of proteins, RNA, and large complexes of macromolecules. Nuclear lamina, a net-like web of protein filaments, or intermediate filaments in animal cells, maintain the shape of the nucleus. Evidence of a nuclear matrix, a framework of protein fibers extending throughout the nuclear interior, also exists. DNA is organized into chromosomes inside the nucleus. One chromosome contains one long DNA molecule associated with many proteins, such as histones. So I'm not sure, okay, going back, the evidence of the nuclear matrix, the way that they wrote it implied that, like, it may or may not exist, but there's evidence of it. So we don't know for sure. They haven't figured it out yet. So I shouldn't say that it exists because we don't know yet. Not enough research there. As I was saying, one chromosome contains one long DNA molecule associated with many proteins, such as histones. Proteins help coil the DNA of each chromosome, reducing the length. Chromatin are the complex of DNA and proteins that make up chromosomes. As a cell prepares to divide, the chromosomes form loops and coil, condensing and thickening enough to be seen under a microscope. Typical humans have 46 chromosomes in its nucleus, and sex cells, eggs and sperm don't count. They have 23 chromosomes. Fruit fly has 8 chromosomes and 4 in the sex cells. In comparison, nucleolus is where the ribosomal rRNA are synthesized from genes in the cell. Proteins from the cytoplasm are assembled with rRNA into large and small subunits of ribosome. Subunits, or parts, exit the nucleus through the nuclear pores into the cytoplasm, where they assemble into a ribosome. Sometimes there are 2 or more nucleoli, depending on the species and the stage of the cell's reproductive cycle or lifespan. Nucleoli play a role in controlling cell division and the lifespan of the cell. Okay, so this is how the DNA turns into a polypeptide. So the nucleus directs protein synthesis by forming messenger RNA, mRNA, that carries info from the DNA. mRNA is then transported to the cytoplasm through nuclear pores, and then ribosomes translate the mRNA's genetic info into the primary structure of a specific polypeptide. Okay, so it's important to note here we have mRNA and we have rRNA. mRNA is specific for creating a polypeptide. rRNA is specific for creating a ribosome. Okay? So now moving forward, ribosomes, protein factories. Ribosomes, which are complexes made of ribosomal RNAs and proteins, are the cellular components that carry out protein synthesis. Note that ribosomes are not membrane-bounded and thus are not considered organelles. Cells with high rates of protein synthesis have particularly large numbers of ribosomes as well as prominent nucleoli, which makes sense given the role of nucleoli in ribosome assembly. For example, a human pancreas cell, which makes many digestive enzymes, have a few million ribosomes. Ribosomes build proteins in two cytoplasmic regions. At any given time, free ribosomes are suspended in the cytosol, while bound ribosomes are attached to the outside of the endoplasmic reticulum or nuclear envelope. Bound and free ribosomes are structurally identical, and ribosomes can play either role at different times. And by this I assume they mean they can be either bound or free at different times. Most of the proteins made on free ribosomes function within the cytosol. Examples are enzymes that catalyze the first steps of sugar breakdown. Bound ribosomes generally make proteins that are destined for insertion into membranes, for packaging within certain organelles such as lysosomes, or for export from the cell, secretion. Cells that specialize in protein secretion, for instance, the cells of the pancreas that secrete digestive enzymes, frequently have high proportion of bound ribosomes. You will learn more about ribosome structure and function in concept 17.4. Okay, so going on to my notes for ribosomes. Ribosomes are made up of ribosomal RNAs and proteins. The purpose is to carry out protein synthesis, not membrane-bound, therefore not considered an organelle. Cells with increased protein synthesis have large numbers of ribosomes and prominent nucleoli. Prominent or distinguished nucleoli, which produce the ribosomes. So the stronger the nucleoli, the more ribosomes exist or are created, and therefore more proteins are synthesized. For example, or an example would be the pancreas cell, which has a few million ribosomes. Protein synthesis occurs in two cytoplasmic regions: free ribosomes in cytosol and bound ribosomes attached to the endoplasmic reticulum or nuclear envelope. And I just want to note here the difference between the smooth endoplasmic reticulum and the rough endoplasmic reticulum are the ribosomes. So bound ribosomes attached to an endoplasmic reticulum makes it the rough endoplasmic reticulum. Free and bound ribosomes are structurally identical, and their roles are interchangeable. Proteins made by free ribosomes function within the cytosol, and proteins made by bound ribosomes are destined for insertion into membranes, for packaging or export outside the cell, secretion. Pancreas cells, that secrete digestive enzymes have a large number of bound ribosomes. Okay? So, that is it for concept 6.3. Hope I helped you guys. Next video we'll be going over concept 6.4. The endomembrane system regulates protein traffic and performs metabolic functions. Okay? So I hope you guys have a lovely day, and I'll see you guys next time. Bye.